I am Professor S. Krupanidhi, heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research, Andhra Pradesh, India, are giving a series of talks, of which the third topic that is being covered now is immune cells, absolute and differential count. In the previous topic 2, we covered immune cells, their attributes, and again their characteristic features. That's what CD markers, cluster of differentiation markers. Now we are seeing the count of immune cells, how to count, because the count plays an important role for assessing the immune potential of an individual and the disease status of an individual. So now we see the animated video of the WBCs, how they are working in an individual. So as I was mentioning to you in the previous topic 2, that these are all called the corpuscles, not the cells, white blood corpuscles. That means they're all moving. They're all moving inside our system. That is what the blood vascular system. So, but not all cells are moving like this. Majority of the cells are static, like liver cells, muscle cells, pancreatic cells, germ cells, they're all static. Only the blood cells are moving. They're also not only moving, there are times chemotactic attaches to the membrane and infiltrate through the blood vessels, so that if there is any inflammation nearby, these cells get infiltrated at the site of the inflammation. That's what you can see, a few of the cells which are rolling in this blood vessel, they get infiltrated. See now, it is getting infiltrated and it goes nearby the places where there is infection, and it engulfs, it devours the bacteria. Like that, the blood cells are doing their function. So bacteria are shown, and they are getting ingested through the pseudopodial extension. That's what we are seeing in this diagram. And now all this is nothing but a dendritic cell. The dendritic cell is the best antigen presenter cell. It engulfs and processes through proteolysis and presents the peptides of this bacteria to the next communicating cell. That's what T helper cell, T cytotoxic cell. Next. So the objectives of the topic 3 is the quantification of WBCs, to understand the procedure for differential count of WBC. This is most important, that is being practiced in all clinical laboratories. And to appoint symptoms due to the abnormalities in this white blood corpuscles. So after having gone through the topic 3 PPTs that are being shown now, the outcomes that we are expecting from the learners are: the total and differential count of WBCs will be known, absolute and relative counts of each cell will be known, there is quantitative and qualitative WBCs, abnormalities will be evaluated, the terms like leukocytosis and leukopenia will be known. Nowadays, the leukopenia term is being widely used in this coronavirus pandemic. Now let us see the WBC numbers. So the WBC are most important for the clinical assessment of an individual. That's why the doctors look at WBC numbers to assess our health. If number goes up, there is some kind of infection and surgery might be needed, as doctor thinks. Clinics will count the number of WBCs in blood samples. This is called the differential count. A decrease in the number of WBCs is called leukopenia. An increase in the number of WBCs is called leukocytosis. Okay, so WBCs count, normal count. A newborn will be having, newborn will be having between 9 and 30, 10 to the power of 3 per ml. So an adult, a child and adult will be having 4.8 to 10.8, that is the normal values, into 10 to the power of 3 per ml. So this is the normal value which we have to remember. That's what I have been telling to you. These lectures are both semi-technical and semi-popular, so that other than biotechnology students also can follow this. So this is a number we all have to remember, approximately around 5 to 10, 10 to the power of 3 per ml is the normal range of the WBCs in our blood. Now, after having known, it's not only one, WBC is not only one type of the cell, there are nearly 6 to 7 types of the cells that we are seeing now. They are basically classified into leukocytes and lymphocytes. Okay, so leukocytes are 70% of the total population of WBCs. This is almost fixed, and lymphocytes are around 30% of the local population of the WBCs. Okay, of this 70%, neutrophils constitute 62%, eosinophils constitute 2.3, basophils constitute 0.4, and monocytes constitute 5%. So here the interesting feature is neutrophils are predominant among the leukocytes. Now let us see the lymphocytes. Lymphocytes are 30%, of which T helper cells are predominant. T helper cells, as I was mentioned to you, T cell is the master immune cell. So this regulates the total immune function. It is about 46% of the 30%. And B cells are again those are the cells which maintain the memory of our infections. Okay. So now the eosinophils and basophil number is the least. See here, 0.4%, 2 to 3%. Why that much least is? These are the cells which cause allergy. That's what the nature has made them in such a way that their number is the least. But the neutrophils, monocytes, they help a lot in the scavenging, in attacking, and again in antigen presentation. Next, T helper cells are the maximum in number, 46%, and B cells are around 23%. So these are all the differential count. If doctor wants, whether we are having granulocytosis, then doctor will go through the leukocytosis. And if you are having any other type of the blood cancer, all those things, doctor will look into this this lymphocyte population. Okay. Now, leukocytosis. Leukocytosis is a condition characterized by an elevated number of WBCs in the blood cell. This is due to bacterial infections such as appendicitis, tonsillitis, ulcers, and urinary tract infection, leukemia, pregnancy, and hemolytic diseases of newborn, and even soon after the exercise, and emotional stress and food intake. So these are all the conditions which will make the increase in the blood cells in our serum and the blood. Okay, so a few of those things, particularly bacterial infection, certainly causes the increase because the recruitment of cells are there during the type of the infection. Because to avoid the infection, number of blood cells have to get increased. And leukopenia is a condition characterized by a decrease in number of white cells in the blood. So the decrease in number of white blood cells is due to the viral infections. Now people are saying even COVID-19 causes a decrease in the number of WBCs. That's a leukopenia situation. Now some bacterial infections such as typhoid fever reduces, typhus fever reduces, rheumatoid arthritis reduces, and autoimmune disorder called systemic lupus erythematosus also decreases the number of WBCs, and certain regimens such as radiotherapy. So the people who undergo this radiotherapy and chemotherapy for cancers also causes the reduction in the blood cells. This is what so particularly in COVID-19, the doctors are advising that there is a decrease in the blood cell count, leukopenia. So see, simply we can't say the count just like that increase or decrease. There is a procedure how to evaluate the blood cell count. That is through the hemocytometer. So we are going to see the animated video how to go through, evaluate the hemocyte count. So the blood we used to take in a hemocytometer, that is a very thick slide. That thick slide will be having a standard weight, having this cover slip, and between the cover slip and the grid, there is a grid on the slide. The blood solution which is mixed with the trypan blue is going to be kept, and this spreads above the slide as per the weight of this what is it called, cover slip. This is what the trypan blue. So trypan blue is being used because there are few cells which are viable cells. There are few cells which are non-viable cells. Non-viable cells take the trypan blue strain. So there is no meaning of having these non-viable cells getting counted. So that's what here. This is the way. This grid is there on either side of the slide. On both the slides, you can fill up the solution, mix it with the blood solution, mix it with the trypan blue. Okay, so there is a dilution factor involved here. All those dilution factor will take into consideration at the time of the calculations. This we see in the normal microscope around 20 or 40 magnification. 40 magnification. So here, once you see the grid, there is a bigger, what is it called, the cells, and these are all the smaller cells in this hemocytometer. So usually we take the bigger chambers for counting these WBCs. Okay, so for counting the WBCs, see here they are now, instead of manually counting, it is with the fingers, it is being operated through the machine. Okay, so here in each grid there are about 16 chambers, and we are now counting these blood cells, WBCs. These are all the WBCs here, and they are all the viable cells. Viable cells are shown, and there is none of the cell is shown as non-viable. Like this, we can keep counting the cells in this particular what is called the grid. Like this, there is 1, 2, 3, 4 grids are there. In all these 4 grids, we do the counting. Once that counting gets over, then we go for the calculation purposes. So this is what that is shown here is a non-viable cell. So this non-viable cell, what does it do? It takes the stain sooner. The dead cell takes the stain sooner than the live cell. So that's what it is, a dead cell that is taking the stain. This we should not get mixed up with as if that it is a live cell. So it has to be it's a non-viable cell. Okay. Next. So like this whatever we have seen in the video, that's what these are all the chambers that we are seeing here. They are all bigger, what is it called, dimension chambers than that of the small dimension chambers. These small dimension chambers are for counting the RBC, red blood cells, red blood corpuscles. These are for counting the white chambers, are counting the WBCs, white blood corpuscles. Here we can count the cells like this, and we can start moving in this way. Like that, you can count the WBCs. So here our interest is to count the WBCs. In the beginning slides itself, I was mentioning to you, RBC is no longer coming in the immune system. RBC is only for percent saturation of the oxygen. For anemic situation, for healthy situation, see RBC is being used, but WBC is only for immunological monitoring. In this way, we used to have the count. So for an adult, male or female, for a normal reference range is between 4,000 to 11,000 cubic millicubes. So white blood cells count, total WBCs, differential count gives the relative percentage of each white blood cells. That's what we have seen earlier. The leukocytes are 60% and the lymphocytes are around 30%. The remaining cells are there in between. So that's what here we should have the differential count, later the absolute count. As a result, we will be knowing the exact number of cells, the B cells and the T cells also here. Nowadays, people are all going for facts, and that is what the fluorescence assisted cell sorter. Now this is all the manual you can do in the. So in the previous animated video, we have gone through how the blood cells, WBCs, are being counted using hemocytometer. So once the total count becomes 6,000 cubic millimeters, as the neutrophil percentage is 62%, we have to multiply this with 62 and divide by 100. We will be getting absolute neutrophil count as 3,720 per cubic millimeter. Okay. Disorders of cell number. Leukocytosis, I was mentioning to you, an increase in number of leukocytes in the peripheral blood is above 11,000 cubic millimeter. If the blood count is shown, that is said to be, there is an increase in the blood count. That is, because increase in the blood count means why there is increase? It is recruitment of more number of cells into the system. Why there is more number of cells required? So the system is made such that the more number of cells are recruited because the system is encountering an infection. So that's what here. So then that's what one probable answer here. Then the next one is leukopenia, where they reduce white blood cells. Sometimes the infection causes reduction in the number, as we see in the case of the COVID-19. So some WBCs count determines the state of health. Count of total WBCs among infants, children, adults is known. Differential count of leukocytes and lymphocytes is known. Use of hemocytometry is acquainted. Calculation of absolute neutrophil count is known as a typical example. The condition leading to leukocytosis and leukopenia are elucidated. Study questions: what are the total count of WBCs? What is the normal WBCs and differential count values? What are the leukopenia and leukocytosis? How do you calculate absolute neutrophilic count? The total WBCs count of 6,000. So with this, I wish you all to go through the slides and we'll go to the next topic next time, topic 4. Thank you.